Good evening, everybody, and welcome to the Stanley Ho Memorial Lecture. My name is Francis Sella, and I'm honored to introduce the talk to you tonight. Sir Charles Godfrey and Professor Anneke Lucassen couldn't be here, so I'm standing in for them. I was one of the original founders of the Center for Personalized Medicine, and I'm also affiliated with the Oxford Martin School because we share a grant together with Professor Zoltan Molnar and Professor Hagen Bailey, who are in the audience somewhere. And I would just like to thank both St. Anne's and the Center for Personalized Medicine and the Oxford Martin School for co-hosting this wonderful lecture by our esteemed colleague, Jeff Macklis. I'd especially like to thank Clara and Thea and Kath for having organized everything. One of the great aspects of the Oxford Martin School is that they allow you to invite distinguished scholars from abroad or even from the UK. And we have a grant with Hagen and with Zoltan where we are trying to, where we are 3D printing cortical tissues. And in thinking about who we could possibly invite to be a visiting scholar, once I heard about this scheme, I immediately thought of Jeff. I thought Jeff Macklis is really one of the forefathers of understanding transcriptional regulation of not only how cerebral cortex cells make fate choices, but also how their axons find their way to the appropriate target and wire appropriately. So Jeff, almost more than anybody for the past several decades, has taught us about how the brain is built and how these choices are made. So Jeff, welcome. I'm very glad that you accepted the invitation. I'm also very, very happy that your wife and partner, Carol Levin, who is in the audience, accepted the invitation. It'll be a great 5 months spent with you guys. So without further ado, I'll just say just one more thank you, and that is to the Stanley Ho Development Medical Foundation, who have been absolutely instrumental in pushing the ideas and the ethos of the Center for Personalized Medicine forward. So thank you very much.
Welcome to the Oxford Center for Personalized Medicine's lecture by Oxford Martin School visiting fellow Professor Jeffrey Macklis. This is the Dr. Stanley Ho Memorial Lecture, and it is also an Oxford Martin School lecture. I am Ian Hewen, a trustee of the Dr. Stanley Ho Medical Development Foundation and member of the CPM's External Advisory Board. The foundation aims to enhance the quality of medical service and delivery in Macau, Hong Kong, and other regions of China by entering into academic partnerships with relevant worldwide organizations. It is our pleasure to have supported the Oxford Center for Personalized Medicine since 2013. Dr. Stanley Ho passed away in Hong Kong at the age of 98 in May 2020. He was an extraordinary leader and a great philanthropist, and his legacy is his extensive altruistic efforts. The foundation honors Dr. Ho's memory by continuing his spirit of serving our communities and by supporting this annual memorial lecture. The University of Oxford's Center for Personalized Medicine is based at St. Anne's College and the Wellcome Center for Human Genetics, United Kingdom. Its principal aim is to explore personalized medicine from a range of perspectives. Dr. Ho's support for the center marks his vision and commitment to help transfer knowledge of personalized medicine to healthcare professionals and students worldwide. The Oxford Martin School brings together a vibrant community of scholars drawn from across the University of Oxford. Its research tackles issues of global significance with teams working towards real-world impact, from innovations in science, medicine, and technology through to expert advice and policy recommendations. We are delighted that tonight's lecture is in collaboration with the Oxford Martin School. It gives me great pleasure to introduce tonight's speaker, Professor Jeffrey Macklis. The title of his lecture is Beyond Personalized and Towards Circuit-Customized Medicine, Targeting Molecular Address Codes for Diverse Functional Neuron Subtypes in the Brain. Professor Macklis is the Max and N. Wine Professor of Life Sciences and Professor of Stem Cell and Regenerative Biology at Harvard University and Professor of Neurology at Harvard Medical School. He is Director of Graduate Studies in the Department of Stem Cell and Regenerative Biology and co-director of the Developmental and Regenerative Biology PhD program at Harvard University. His lab is directed toward both understanding molecular controls and mechanisms over neuron subtype specification, development, diversity, axon guidance circuit formation, and degeneration in the cerebral cortex, and applying developmental controls toward both brain and spinal cord regeneration and directed differentiation for in vitro therapeutic and mechanistic screening. Professor Macklis, it is my delight to welcome you.
So thank you all for coming. Thank you, Clara and Thea and Catherine, and thank you, Francis, for such a nice introduction, although I always like to correct the introduction if I can find an error. And I haven't been active for several decades. Come on, 2 to 3, let's say. So a couple, few. But other than that, thank you very much for the generosity. What I didn't ask in advance is whether I have a laser pointer, but I think I do. And what I'd like to talk with you about tonight is something somewhat daunting for me because it's not a topic with which I've engaged directly. I've had some thoughts over time, and when the Oxford Martin School and the Center for Personalized Medicine asked me to give this important lecture in Stanley Ho's honor, I thought that I would try out some thoughts with you and just share them for the first time. So I've put together a lecture where I'd like to go not above and beyond and more important, but beyond personalized medicine, and I'll explain what that means by the NHS, and toward what I've just termed, and I made this up for this talk, circuit-customized medicine, thinking about every individual type of circuit and neuron that goes wrong in human diseases as individuals with individual biology. And to use what I'll put together for you in broad terms as molecular address codes to try to think about sending biomolecules and therapeutic agents to distinct classes or individual functional neuron subtypes in the brain. And I'm going to explain all of that to you. And let me just point out, brain, human, cutaway, cerebral cortex, I'll explain more in a minute or two. Some imaging from MRI of humans of large axon tracts and bundles, just the sort of the major highways and throughways of the brain. And I'm going to tell you mostly about data that come from mouse as experimental model that has a much smaller cerebral cortex, but with essentially all the bits and pieces, the same organization, just much smaller in area. So first, and most importantly, I want to give deep thanks to the Stanley Ho Memorial Development Foundation for setting up and funding the CPM and making this lecture happen. To the Oxford Martin School, with their generosity, who've made my entire 5-month visit happen and let me engage with my colleagues I'll get to, and for giving a broad intellectual environment that I've already been able to join in. The Center for Personalized Medicine, for obvious reasons, but also St. Anne's College and Plumer Fellowship that's given me a college home that's really been a nice academic home, and St. John's College that's provided my wife Carol Levin and me a place to live and visit and take part in wonderful fellow activities. The Department of Physiology, Anatomy, and Genetics for giving me a departmental academic home, and very importantly to me, my longstanding friend and colleague Francis Zella, my close, long-standing colleague over 2 decades or close to 3 in cortical development, Zoltan Molnar, and my more recent colleague, and now that we've had enough beers together, friend, Hagen Bailey, where we can brainstorm and talk about ideas. And I first met Hagen only by Zoom during the pandemic, probably 2 years ago, when we were planning this visit. So here's personalized medicine from the NHS, directly from the website. Imagine being able to get a faster diagnosis of a condition based on your unique situation. Be given personalized treatment, not the broad cancer treatment, the broad diabetes treatment, the broad cardiovascular treatment, based on what would be most effective for you, your genes, your polygenetics, your underlying conditions, and experience fewer side effects, or even move away from simply managing to predict certain conditions and potentially prevent them from developing in the first place. And this is the basis for personalized medicine and understanding the role our DNA, and the reason I stress that is I'm going to move away from DNA, plays in our health, might be able to transform how we think about healthcare, predict, prevent, do more precise, and personalized and targeted therapeutics and interventions. So I've translated that to my way of thinking about the title that I told you about, towards circum-customized medicine and targeting molecular address codes of neuron subtypes in the brain. So circuit-customized medicine, and I coined this term for this lecture, so I'm floating this idea with you here. Imagine being able to better delineate diagnosis of a condition based on the unique biology of certain specific brain circuits that undergo pathology, and be given circuit-customized treatments rather than a small blue pill that affects hundreds, if not thousands, of kinds of neurons and circuits in the brain because they share the same neurotransmitter or the same signaling pathway. And potentially selectively alter that circuitry, et cetera, about fewer side effects, promoting health. And maybe this could form the basis for circuit-customized medicine. By understanding our subcellular, I'll get back to that, not just what's in a cell, but where is it placed in an incredibly complex neuron, especially its RNA, I'll explain, and protein molecular machinery employed by specific neuron types. And in my lecture tonight, I'm going to show you new evidence, some of it published, some of it unpublished, that individual circuit-type neurons in the brain use RNAs and proteins quite distinctly, and that gives some avenues, some twinkle in my eye, that we could target those to be much more precise in therapeutics. And that might get to circuit and dysfunction customized and targeted interventions. So I'm going to try to show you some faces along the way, but this is my lab group as we came out of the pandemic. Harvard's not all red brick. I'll show you a few pictures later. They're a great group. I won't be able to give full credit to each of them and to each of the people's work not in my lab who we've taken and built upon, but I'm going to try a little bit along the way. So back to that brain, nose, eye, skull is cut away. This is the cerebral cortex. That's the cerebellum. Here's the brainstem, and it extends down to the spinal cord encased in bone. No tests at the end, but I've designed this lecture thinking that some of you are much more expert geneticists than I am, and some of you might be economists or historians or sustainable agriculture folks. So I'm going to try to tap on all of those and make everybody somewhat happy, but probably not please anybody entirely, but that's okay. I'm going to, because of reasons I'm going to get into, I'm going to focus more on the broad with a few tidbits for the more esoteric based. And that's because, and I'll tell you more later, I'm giving 2 talks over the spring for the specialist audience. So let me give you my view, and I don't think it's a very controversial view, but I've put it in specific words, that the central function of the brain and spinal cord, together those are the central nervous system, or CNS, and the cerebral cortex in particular, what most of us think of as the seat of our higher cognitive function, our language, our thought, our intellect, our planning, what we're using for this lecture, is to process and integrate input sensory information into motor output. Now that's not just violining and dancing and surgery and learning to use pincer grasp and fine calligraphy, but it's also my elocution, my speaking, my moving through the world. But it's sensory input to motor output based on experience and optimally with cognition, choice, and benefit. That's complicated. So again, I'm going to take you from this brain and its complicated circuitry, these are just the major highways, and deal with something that's been a long-standing question in neuroscience of the, quote, sorting problem. How can there be literally 85 billion neurons in the cerebral cortex, each with between 1,000 and 10,000 synapses, each with one to a few axons that I'll explain to you connecting, and how do they not get all jumbled up? And even if all of our brains are not exactly the same, how are we so amazingly similar to read history or Proust or philosophy and communicate and use metaphor or poetry? And I'm going to try to dig into the very superficial aspects of that problem. And again, I'm going to show you data largely from mice. So, there are going to be 4 parts of my talk this evening over the next 40 minutes. And those are a brief background about the cerebral cortex, neurons, and the wonders of diverse circuitry. Then a few brief vignettes about a few relatively recent discoveries about how neurons function that set a foundation for the concept of this circuit-customized medicine I've already explored with you and set out for you. Then a quick peek at subcellular molecular biology of diverse functional circuitry of the brain. Then at the end, in a little 2-part 4, recent work and work in progress toward understanding human disease and subcellular molecular machinery. And there I'll turn a bit of a corner into human biology. What I'm not going to show you here is images like this. You know, it's late, there are drinks coming. Most of you are not card-carrying molecular developmental neurobiologists, or maybe some of you are not, so I'm not going to do that. But for those aficionados, here are the 2 upcoming talks by me with more biological depth. There's going to be what's a relatively broad biological science talk at the end of April, and in the coming few weeks, I'll be giving, and that'll be the Department of Physiology, Anatomy, Genetics Head of Department seminar. And then the Oxford Cortex Club, Monday, February 13th at 4 o'clock in the department, but in a more esoteric for the aficionado way. So part 1. Remember, that's the brief background about cortex, neurons, and the wonders of diverse circuitry. So here's the patron saint of all of neuroscience, Santiago Ramon y Cajal, poor Spaniard from the north, wasn't accepted, trained as a surgeon barber, later became a physician, got a Zeiss microscope, brass in those days, a lot of roadkill, and discovered out of first principles by looking through a microscope that I've used the same model number at Harvard from a collection of historical instruments that Alex Agassiz used. They were bought in the same year, within a year or 2. It's unbelievable what he discovered with that instrument. And what he discovered So first, he didn't discover this, but the cerebral cortex that I showed you is about the size, you might have been wondering, of a large dinner napkin spread out. That's what it is. And it's crunched into our head. It's about 2 millimeters thick, more or less. So it's basically a thick styrofoam pad. And it's crunched into all those sulci and gyri in order to fit in a skull that can be small enough at birth to fit through the birth canal. So it does wacky and wondrous things to make itself more complex from mouse to non-human primate to man. One thing it does is it takes so-called pyramidal neurons, and Santiago Ramon y Cajal was awarded the Nobel Prize in Physiology or Medicine in 1906, one of the first several years of the Nobel Prize, for the identification of neurons. Before that, it was thought that the whole nervous system was a big spider web, syncytium. It was all connected together. Because how could this network of all this gamish be actual individual cells? And he, using that lousy Zeiss microscope, identified entire neurons, their cell body, their basal dendrites, their apical dendrites, where they placed them. But what he also showed was that across the cerebral cortex, especially as we get up to non-human primate and man, but in mouse in a big way too, cerebral cortex, that 2 millimeters thick, varies within the space of a human hair. It can be 100 microns, and it can shift from that to that with different proportions of distinct types of neurons, still in the same 6-layered structure, 6, 5, 4, 3, 2, 1, but with very different cellular proportions, types, and he presumed circuits and functions. And that's really quite remarkable, and I'm going to get into that. Now, for those of you who are not really biologists at all, let me go way back to the beginning and just tell you neurons are really cool, unique, and complex cells. These are red blood cells, and these are at scale. I made these about the size of the nucleus where the DNA is of this neuron, because that's about how big it is. 8 microns, 10 microns. Here's the nucleus of this cell, and this is all, everything brown is a single neuron in the striatum, with its basal dendrites, its apical dendrites, its axon coming out down here and going out of the plane of field. That's what we need to contend with, because each nervous system disorder affects distinct types, classes, or broad or narrow, or singles of neuron types. So here's one example, corticospinal neurons, CSN, that give us, they're the only output from the brain to our spinal cord, and they provide all the fine dexterous motor movement. They let me speak and go like that with my mouth and my face and wink and twiddle buttons. That's the corticospinal system. It doesn't exist below rodents in any other kinds of animals, and rodents use it for fine dexterous use of their forelimbs. It turns out these neurons selectively degenerate along with spinal motor neurons in the spinal cord in motor neuron disease, or ALS, or what we term in the United States sometimes ALS or Lou Gehrig's disease, a great New York Yankees baseball player of the 1930s who within 6 months went from being the best batter, fastest runner, all around great, to barely being able to limp to first base, and then he retired. And ALS is a devastating, essentially always fatal within a few years disease, but it has a set of mutations that account for 10 or 20% that are in every cell in the body and in every neuron type in the brain. But only these 2 kinds of neurons degenerate centrally. How could that be? That's what I want to explore with you. Now, here are 3 of those corticospinal neurons, and I blew up and didn't expand, I just blew up Cajal's drawing, same proportions. That's how close he got to right with that brass microscope. It's astounding. And he called these pyramidal neurons the noble and enigmatic cell of thought. He was quite poetic in many of his writings. So here's another kind of projection or pyramidal neuron. This is before neurons were discovered. This is a human brain splayed open from below. And these are literally hundreds of millions of parallel aligned axons of neurons that look like this. They're called callosal projection neurons, and precision of all this circuitry and connections makes us sense the world, think, and move. And these noble and enigmatic cells of thought, let me tell you, you might wonder why fist and body. If this neuron, and it normally is about the size of a fist, its axon going out there would extend 2 to 3 up to 7 kilometers with precision and find a target about as big as this screen with that much precision. Astounding across the city of Oxford. If it was the size of my body, its axon would be the size of my arm in diameter, and it would extend 20 to 30 to 70 kilometers to Heathrow Airport and find a target the size of this room, about 10 times my body length. Astounding, with precision, with all the twists and turns. And each type of neuron does that distinctly. Therefore, the sorting problem. And the thing is, these neurons don't just find that target, then they make synapses with precision. And I'll tell you about that in just a minute. So here are some neurons in cell culture on a dish. They share a lot of those characteristics, but they really just don't capture the full flavor of neurons that are in the brain. Because they look like this, they have their cell body. Within a day or two, they'll extend an axon. At the end, they have something that's called a growth cone. Sort of looks like an extended hand. It has touchy-feely bits at the end that sense the environment and make all the decisions to get it to Heathrow Airport. The problem is, and here's one in vivo with much more complexity than here, sending its axon within the first couple of days, and there is its growth cone. And each of these types of neurons that I've told you about, and if we're lumpers, there are probably a couple of hundred types. If we're splitters like I am, probably 1,000 to 10,000 types with distinct circuits. They're born from progenitors or stem cells, these beige guys that I won't get into, and migrate up into this 2 millimeters thick plane of the cortex and make diverse subtypes. So they're not all the same within a layer 6 or 5 or 4, and they send their projections. And I won't bore you all with this, but these are images as if they were a salami slice this way through my brain, coronal. And all the little triangles, the pyramids, are the cell bodies sending their color-coded axons in these schema to either targets on the other hemisphere of the brain. This is one side, that's the other side. Or targets forward or back between visual and somatosensory or auditory systems. So we can smell chocolate and remember a fond memory and know what it looks like. It connects all those modalities. These guys send output to the spinal cord or to the thalamus to control motor and sensory integration. And there are even a number of neurons, more every year, that have multiple projections that don't just go to Heathrow, but connect to Heathrow and connect to another side of Oxford and send the same information or distinct information. And now let me just tell you, my arm, if I extended my arm to Heathrow in your imagination, the volume of my arm, or the volume of an axon of most of these neurons, is upwards of 95% of the volume of the cell. Yet in neurobiology, we've almost always only thought about the cell body. And I'm going to tell you about new approaches that we developed over the past several years to look in a subcellular way at distinct types of neurons. Now, the good news is many investigators, my lab among them, but not leading this, have identified over the last 50 years when neuron types are born. And we can use that cleverly in experiments that I won't go into because of this audience to target classes of neurons and then target in other ways to get to subtypes with precision. Now, here's the first breather, because some of you are getting really scared. But here's the problem: science versus everything else. So everybody's online for the simple but wrong direction. And I'm going to take you down the long and windy road of complex but right. And unfortunately, in the nervous system, I think to deal with these major issues with precision, we're going to have to do the complex but right. So here's part 2. A couple of brief vignettes about some recent discoveries about how neurons function that set a foundation for this circuit-customized concept. So, just a refresher for all of you historians and economists in the group. This is what's called the central dogma. It's not a political party dogma, but it's the central dogma that DNA that resides in the nuclei of our cells has the code of life. It's built to be really well maintained. It has error correction. It's like the hard drive, permanent archive. But through a process called transcription, it's turned into very closely related, but not exactly the same, RNA. RNA carries the messages. It's almost like the USB drive that goes out to the machining tool or the something. It's the portable thing that can make, through what's called translation, protein. Those are the working nuts and bolts of cells. Those are real functioning things, proteins. And this is a somewhat science fiction view of a nucleus and the DNA within it making mRNA or messenger RNA. And I'll show you bits and pieces of this through my talk that engages with 2 parts of a ribosome and spits out proteins that form these twists and wiggles that make things like meat or muscle or a peptide or other things that get coded for in the DNA. So I'm not going to take you through this, but over the last 20 couple of years, my lab and others have identified what I would view as a combinatorial, meaning combination of multiple instructions together, not a single one at once, molecular logic for cortical neuron development. And that goes from where the progenitors sit, green progenitors versus red stripe ones, out of those progenitors, whether they make the purple class or the golden class of cells, those golden class of cells, whether they get repressed in a certain way to make neurons that are Professor Molnar's long-standing favorite of the subplate, or corticostriatum, or corticothalamic, or corticospinal neurons, and then build that 6-layer cortex. But Cajal was 120 years ahead of us because he pointed out this thing about the aerial development. So my lab and others have also spent a lot of time investigating the diversity across primary functional areas, motor, somatosensory, auditory, visual, and we've identified, along with others, a number of molecules, it's getting scary again, that are here in these outlines that control these aerial functional units, like sensory versus motor, visual versus touch, and control the circuitry in those units. So really that dinner napkin of a human brain is probably hundreds of distinct little cerebral cortexes stitched together like in a big mosaic quilt. And they function like little cerebral cortices. They're similar, but distinct. And I think if we want to really think about human disease, we want to think about them with precision. So I spent a little too much time at MIT in my younger years, and it always came to me that this combinatorial set of transcription factors, proteins that bind to DNA and control which RNAs are made, form a nested semi-Boolean, for George Boole, molecular logic for making distinct types of neurons, their diversity, their circuitry. I would propose their evolutionary expansion into our amazing brains, but also what goes wrong with selective types of neurons in degeneration, and how we might turn this around toward directed regeneration. So I'm not going to tell you this whole story now. I just wanted to show you a beautiful brain. This is a mouse brain, again, a coronal section from one side over, with distinct types of neurons labeled with molecular labels that have all been identified to control their birth and development. And I am going to get back later, when I get to human disease, to work of Molly Woodworth and Luciano Cousteau-Grieg from a few papers several years ago, about this molecule now by consensus termed BCL11A, that sets up that Santiago Ramon y Cajal balance across areas of the proportions of different kinds of neurons, and sets up the organization of all the sensory maps of the mouse brain, and we think the human brain probably as well. And the reason we think that is we postulated that if something went wrong with this gene, it would really mess up the human brain. And over a few years' time, 4 genes on a chromosome were identified where a mutation existed that caused a human disorder of intellectual disability and autism. Then folks narrowed it down to 3 genes, then 2 genes, and now it turns out that there are, I'll tell you about that later, 20-some mutations in BCL11A who monogenically, with essentially full penetrance, meaning you have the gene mutation, you get the disease, of human childhood autism and intellectual disability. And I'm going to get back to some work on that. So here I'm going to go much, much quicker. This is like a tasty tidbit for the aficionados. And Helen King said something very nice on Friday night at a reception that I'm going to be giving a trailer for my other 2 talks. So these are a couple of the action scenes that I'll tell you much more about work from Vibhu Sani, Sarah Schneider, and Yasu Ito about molecular controls over CSN segmental specificity, meaning controlling my face, my arms, my thorax, my legs. That's all precisely wired, or else we would just sort of wriggle around in a ball. So the spinal cord is organized very segmentally. It has connections from the cerebral cortex. The ones that control face and forelimbs sit far on the side of the cortex. Here's the front, the back of the brain, the bottom, the top. The ones that control my legs are up near the crown and over into the center, and we wanted to find out what molecularly makes that happen. And that heterogeneity is not just relevant, for example, to idle curiosity, but to disease, to motor neuron disease I've already told you about, because some forms of ALS affect only the face. Some affect essentially only the legs. So it's not a matter of length of neurons, it's molecular specificity of circuits. I'd like to come up with some ideas to target those populations with more precision. So, long story short, we identified, it's been long known what I already told you, that those that go to the bulbocervical regions, meaning the brain stem and cervical spinal cord, here's a mouse brain from the top down, cerebellum, olfactory bulbs, the eyes would be there. Here's the cerebral cortex. You notice It doesn't have all the crinkles and valleys and hills. It's a non-sulcated cortex. It's lissencephalic, smooth for you classics folks, and it's much smaller. It's only about the size of my pinky last knuckle opened up. So it's more like a centimeter squared rather than 20 inches square. But it has little areas, not as much complexity. Mice don't see as much facial recognition and precision as we do, but it's organized the same way. So it was known that their bulbocervicals are further rostro toward the front and lateral toward the side, and those that control the legs are more caudo toward the back and medial toward the middle. But what we discovered is that in fact, tucked in here are two totally distinct populations of neurons. There's what we believe to be an evolutionarily older bulbar cervical control set that also sends projections, but with less specificity to the bulb and cervical area. And if we leave them alone to do the work, mice can't do this. They can only do this. And we're interested in how did maybe these guys take over more function from those guys. So in, I think, 3 slides, I'm going to tell you that we first identified there are some specific molecules, whether they express or make the RNA for the molecule or block it, that set up each of these 3 populations with precision. From the moment they're born, they know who they're going to be. They make different projections. They make different kinds of target innervation. And further, the new evolutionary green guys actually secrete a molecule. This is a whole, what some Twitter commenters said, is a new mode of axon guidance. They secrete something called lumacan that was previously never known to be made by neurons, and put it out. These are supposed to be protein molecules blown up, millions and millions of times. They secrete it where they would like to make connections. And what that does is it blocks the blue evolutionarily older guys, and it blocks the guys that want to control the legs. It's almost like a dog or a fox urinating around territory. Just stay away from here. It's not entirely effective, but it's about 75–80% exclusionary of those other populations. If we take it away, those other populations dig in, and the whole 4-limb behavior is wacky and not very good. So we put this forward as maybe one exemplar of how evolution can have a new population and diverse type of neuron find its way in and exclude other types of neurons so it can do a better function. And just so you know, this isn't totally irrelevant, I think, here is marmoset brain, and human looks very similar. Here's lumacan in 2,000 neurons out of 100 million or so. And here's lumacan extending through this whole part of only lateral, again, motor cortex in this non-human primate. And of interest, a couple of mutations in lumacan have been identified in one subset form of motor neuron disease, ALS. Okay, so that's even more complicated, but please bear with me. We're about where that guy is. Long and winding, part 3 of 4. So back to all of this complexity. And I'm going to give you just a quick peek, broadly and for the aficionado, about what I mean by subcellular molecular machinery of this diverse functional circuitry of the brain. So many people have likened this kind of amazingly complex human circuitry seen by MRI with a little bit of guesswork into this sorting problem. And in some places in the nervous system, it looks like a bit of a, old-style PDP-11 rack with wires going everywhere. But in other parts of the nervous system, it's tightly fasciculated and immensely organized. And there are different mechanisms that have different types of circuit neurons do different things. And frequently, the more evolutionarily advanced neurons organize themselves this way. So here's where I'm going to tell you about those growth cones. This was Cajal's drawing, or one of them, from about 1880, from developing chick. He got eggs, grew them, and looked at the chicks with very crude methods and intuited a timeline. He had no time-lapse video microscopy, that a neuron starts off like this, a round ball cell. It extends an axon. It has what he termed, and he coined it here, a protoplasmic motile cone of force. Maybe I put the adjectives in the wrong order, but pretty much that. Growth cones, termed them, and then he showed they keep on growing and growing as the neuron is sending out its dendrites. He pointed out that there are what are termed collaterals on many types of neurons, but amazingly, not only did he first identify that there are synapses or breaks between one neuron and the other, and he posited there must be chemicals put out by one to go to the other, and he posited the direction of the circuitry all with a brass microscope from 1880, but he also said these growth cones mature into synapses. So everything I'm going to tell you about in growth cone biology, we're actually very interested is actually the setting up of functional synapses. So here's the problem. Neurons are uniquely polarized cells, and they have problematic biology. This is a cartoon of an average liver or kidney cell, and it has its nucleus, and it has its DNA, and all of the functions happen within the confines of that cell. And that cell might be 15 microns across, a seventh of a human hair in diameter, but it's all right there, like a ball, like that fist. Here's a neuron from a textbook. It has its cell body, its nucleus, its axon that's encapsulated in myelin for conduction velocity by insulating it. This, I found out, was what our third grade daughter must have copied this from for a third grade science project. It wasn't from me. But this is a very false impression, because if I shrink this down, you'll see, to this size, and match it to the cell body size of a projection neuron, here is the axon of that neuron. It's like an endless customs line at the worst airport you could ever imagine, extending from here to Heathrow Airport. 95+% of its cytoplasmic volume before it makes synapses. That's astounding. Why would a neuron ever want to only respect what's happening back there? It could be a meter away from where it's making a synapse. And we thought that in different subcellular compartments of neurons, there might be quite different molecular machinery. And I'm going to close in the next 10 minutes. So it's also true that the central nervous system doesn't regenerate. Cajal did these experiments with his graduate student, Teo, published them in 1911. Turns out, if one makes a spinal cord injury in a human or in a mouse or a rat or a rabbit, growth cones try to grow, but instead of making beautiful feeling hands, they make these so-called retraction bulbs, dysmorphic growth cones. Why is that? We intuit that maybe the answer to that is in their subcellular molecular machinery, not back in the cell body, because people have looked for decades without much help. This fellow, Llewellyn Barker, in 1899, and I'm going to paraphrase, does the axon and growth cone receive all of its molecular material from the neuron cell body? Or does it do that, maybe a priori much more likely, for the most part, localized, needing only the overall influence of the cell to which it's connected to govern assimilation? And we wanted to explore that area. And Alex Polopoulos, now an assistant professor at University of Maryland, and Alex Murphy, a former graduate student, took on growth cone molecular machinery that we think builds this wiring and later synapse formation in a subtype specific way, its maintenance, function, presumably dysfunction, its longevity, perhaps reasons for neuron degeneration, and directly limits or might be able to enable regeneration. And I'm going to mention some work by some pioneers in related fields. So people thought this guy, when he was much younger, was totally crazy because he took electron microscopes and found that there were ribosomes that make proteins out in the dendrites, the other end of neurons. And people said, no, there are no ribosomes out there. Ozzy, you're an idiot. And that happened for about a decade until he turns out not to be an idiot and everybody else found out he was right. So I want to give him credit. Christine Holt at Cambridge and her incredibly important lab took first steps, and I'm only giving credit to a few here, so I'm sorry I can't spread it more widely, of local translation in axon development, largely using visual retinal ganglion cells, neurons, in culture, and identified that if one blocks translation, growth cones don't move. John Flanagan in the Department of Cell Biology, my friend and colleague at Harvard, found about a decade ago in this paper and since that specific receptors activate local protein synthesis in growth cones and presumably at synapses, saying that there's regulatory control. Aaron Schumann has really been a pioneer in thinking about local dendritic protein synthesis in memory and learning. And Jeff Twiss and Mike Fainsilber, individually and collectively, have used sensory neurons to get at some molecular mechanisms of how specific types of RNAs are sent down axons. So Alex and Alex knew this, and I think because of time, I'm going to skip over some of the things that we thought about. And they wanted to study growth cones not on flat cell culture dishes like this guy moving, but rather in a living brain. And this is a little bit of one of those coronal sections. Here's vaguely seen the outline of an entire mouse cerebral cortex, where we've labeled the cell bodies and nuclei in green using some tricks I won't go into now, and their axons in red, in ways that we can then isolate growth cones. And the way we did that is we came up with ways to pluck off the growth cones from very complicated environment where they're sending axons. And this is an unretouched image. The rest of the brain is just black. So we came up with ways of biophysically shearing them off using some old EM methods, using what's called a sucrose gradient to isolate particles of a certain size, red ones and not red ones, and then use some special approaches similar to FACS sorting to isolate only the subtype-specific red guys and look at their RNAs very deeply and their proteins by mass spectrometry. And this is our cartoon of what it looks like by labeling a specific class of neurons in a specific area. They're sending their axons into this amazingly complicated environment, but we get rid of all the non-red stuff, get rid of all the cables of axons by using the FACS sorter, and get only these growth cones. And what we found, when we do that, is we can quantitatively map where every protein and every RNA is to it's either the cell body or growth cones. And we find, without going into details on this otherwise scary slide, every one of these circles is a distinct protein. So this is the proteome of a specific class of neurons, growth cones, and it's basically all the normal cellular machinery to make proteins, to break them down, to process them, to interact with the environment. And these are a subset of its RNAs. Remember, DNA, RNA, protein. And the blue guys, about 60% of them statistically powerfully, significantly distinct between growth cones and cell bodies. These guys are all involved with a famous pathway that some of you know about, the mTOR pathway. They're also called 5' top mTOR hypersensitive transcripts. That means they share a certain code in their RNA. And we identified, not just at the transcriptomic level, but then we grew neurons of this type in culture and saw that at the beginning of life, this pathway is all in the cell body. During axon elongation, it's trafficked, put out in the growth cone to control. A week later, it's back in the cell body. We can watch this at the protein level. We can watch it. All of its interacting partners are out at the growth cone. We can look at this in so-called single molecule fish, looking at individual RNA molecules labeled by each red dot is an RNA molecule. Hot, hot, hot in these growth cones that Cajal so aptly named years ago. And we can interfere with this process in a particular sort of clever way at the right time of development and totally get rid of connections between the two sides of the brain. So these things and putting molecules where the neuron wants it are critically important. So now, second part of part 3, and I'm going to, I think, go over this so quickly as to be vanishing and just tell you, because I want to get to the last part, that we've identified by using these approaches in callosal neurons, corticothalamic neurons, corticospinal neurons, that there are distinct RNA sets out in growth cones of each type that have some shared machinery but are quite distinct. And those include very powerfully trafficked molecules that are highly enriched in recent human studies of genes that are risk genes for schizophrenia and autism spectrum disorder, but importantly, not for bipolar. So it's not that it's enriched for every disease known to man. And these have to do with synapse formation and circuit building. Now, there are lots of ways for RNAs to get shipped down from a cell body to growth cones, and we're exploring many of those all at once. And we've identified that a series of subtype-specific RNAs, and thus their proteins, that have to do with a receptor-ligand pair for setting up growth cones and specific synapses are subtype-specifically expressed and placed in growth cones of callosal versus corticothalamic versus corticospinal. And I won't get into their receptors, but I will tell you that the way we think we get there, those RNAs get there selectively, is by parts of their RNA code and by specific molecules called RNA binding proteins that are subtype specifically expressed. One type of neuron has this family of RBPs, this family has another, etc. And they ship RNAs where they're needed. And I'm going to skip over that. So central dogma again. And we're now interested in studying this at a very fine level of how translation is regulated. I won't go into this. What the proteins are there, and we've developed ultra-low input proteomic investigation. And we're very interested in what's happening in these retraction bulbs. So now part 4, and I'll try to be very quick. So we took this autism gene, had done an experiment that found some very specific circuit disruption. We knew it's now a human disease gene. Here's the syndrome, here are the mutations. We developed an approach by which we can make half the neurons in a certain spot mutant and half normal. And then we can look, and we turned green, blue into cyan and magenta for those with color distinguishing issues. And we can see that the normal blue control neurons and some of the mutant neurons make connections to the other side. But only the mutant ones, shown here in black, make this dramatically abnormal anterior commissure that normally is tiny like that and only old mammals like platypus have that. Similarly, strikingly dramatically, only the purple mutant guys send axons like a beeline selectively for this basolateral amygdala. The whole amygdala is about that, this specific nucleus. And some of you might know that this is centrally implicated in fear and anxiety and centrally implicated in autism and response to sensory input. We're very intrigued with this. And we can sort their growth cones, and we can find genes that are, for example, too high in the cell body but too low in the growth cone, and then manipulate them and find we disrupt that circuitry. So now, the last 2 slides, and then I'll close. We can now take human stem cell-derived neurons into something called organoids, fuse them together. Here's one of the cerebral cortex, or this one's of the cerebral cortex-like. This one is spinal cord-like. They send axons across. They have growth cones, and we have sorted those growth cones, and we have retrogradely labeled for the aficionado and sorted the human neurons responsible for those, and we can now look at their RNAs and proteins. So in summary of our brief time together, I gave you a bit of a brief background about cortex, neurons, and diverse circuitry, told you a few recent developments, told you a little about subcellular molecular biology, and told you a little bit of recent work about human disease, both in mouse and with human cell talks. And for those of you who'd like to come, here are my next talks, and I'll leave that on at the end. So again, simple but wrong, complex but right. And I love this quote from this physicist philosopher, Emerson McPugh, 1938. If the human brain were so simple that we could understand it, we would be so simple that we couldn't. And I'm going to leave you with that and take you back to this idea of medicine, acknowledgments of the former members of the lab, current funding sources. Thanks again to everybody here at Oxford that's made this possible, sharing a few views of my building on the bioscience campus and our 2 buildings of our department and our quadrangle, Leverett House, where I've been a member of the senior common room for many years, and Leverett and an old postcard from the Charles River. I won't bring this up, but we have lots of ties between our 2 institutions going back to 1929, where there are a number of Oxford physiologists at the Physiological Society. And I'll leave this on for any discussion. Thanks very much, and I'm sorry to go long.
Thank you very much for that wonderful tour de force. It was extremely interesting and enlightening. And I think we can sit down and relax for the questions.
Sounds good.
So everybody, there's about 10 to 15 minutes for questions. We can take questions from the audience, and you can take questions from the online audience as well. So Jeff, I'd like to kick it off. So,
And there's one in the back after you.
Oh, great. So this was an incredibly interesting perspective on very, very long neurons. Do you think there's a threshold within which you have to sort of go beyond the threshold of a certain length before the proteins and the specific transcripts in the growth cone become relevant? So really my question is, what about the short interneurons?
Yeah.
Would they be guided by the same, and at what point
Yeah.
would
So
that shift?
Francis asked, so I'm going to try to be quick in all my answers so that we can get to more questions and more answers. There's a class, the whole other class of neurons, biggest class of neurons, it's a smaller class than projection neurons, but the whole other large class are so-called interneurons. They're more like if the cell body was the size of my body. They would have axons and dendrites that would reach up to the ceiling of this large room. They connect to very particular types of projection neurons. They're largely inhibitory. They modulate, modify, tune, and they are of amazingly beautiful diverse, miraculously diverse morphology, thus thought to be circuit specific, chandelier cells, basket cells, these guys that you know and some of you know. I'm inherently lazy, not really, but we decided to tackle this kind of biology in projection neurons because we could isolate subcellular compartments. But I have no thought at all that the same biology wouldn't be working in interneurons. Let me take away the double negative. I have absolute confidence that those interneurons that are making specific circuitry also ship things to their terminals, because even if you think about this, the lengths, the decisions of shipping different things to my input side, my output side, some interneurons connect with multiple kinds of projection neurons with distinction. So I think it's there, but it's just we're not going there. Is that on target to your? There was a question far in the back.
Yes, please.
Hi, thanks so much for your talk. That was very interesting. So one of the reasons why I wanted to come to this talk was because of the personalised medicine element. And obviously you've shown a huge amount of data, and it just shows the mind-blowing complexity of targeting the brain therapeutically. Do you think that we're anywhere near having the molecular tools to get that kind of delivery, localised and specific, dosing? And obviously the NHS, as we know, is a mess. So in this country, for example, the cost of those therapies is going to be huge. I don't know what it would be like in sort of a more American sort of system, health system, where you're relying on your insurers to pay for it. But it just seems like we're so far away from being able to have that sort of personalised medicine, and also and to regulate it. Again, the whole regulatory side needs to be touched on. But yeah, also having the infrastructure and financial sort of support to deliver them.
Right. So we're just meeting, so we don't know each other, but I'm going to give you an inkling of my personality. I am a combination of a venturesome dreamer and a historian, meaning I like to look back to see what seemed like bonkers 100 years ago, or 50, or 30, and is now everyday practice. So we now, every day for 4 cents or 12 cents or 10p, give out serotonin type 2 receptor uptake blockers. And 30 years ago, that was inconceivable. So when you say anywhere near, I think we're not around the corner, not the end of this decade. I think in little bits and pieces of getting better, but still not so good, but better than a blue pill, and then better, and then better. I think over decades and scores of years, this could become reality. That's my venturesome side. And when I look back over history, we have antibiotics that target things with precision, not just penicillin coming from mold, but if it weren't Fleming saying, I think a lot of molds make a lot of stuff that probably kill a lot of things, we'd be not looking. And those things are now cheap. So that's my view. About the NHS and the US healthcare system, in my view, these do not need to be $100,000 gene therapies. I think they could be more akin, well, I won't lay it all out here, but yeah, I don't see that it would need to be that, if that makes sense. And I think it could be more like the kinds of personalized medicine we use for subtype variants of certain kinds of cancers that used to be lumped just 10 years ago. Does that make any sense? Is that on point?
So I was just going to say, with regards to the timescale, do you really think we'll get there before humanity's destroyed itself?
Well, there I need other venturesome predictors in the audience who, and we need to do a number of things to try to make those curves not cross. But I can't address everything in humanity.
There was a question in the back, and I'd like to encourage the online audience also to participate in the questioning. Just send your questions via chat.
Yeah, it seems like there's one.
So it's just using the Q&A button online, not the chat, for the questions. Thanks.
Should I? How about in black and then in purple?
Thank you. I'm wondering, so on the therapeutic front of maybe specifically targeting the circuit-based neuronal type, do you envisage a platform-based technology where you have a discovery program where you can identify systematically the targets within each subtype and then develop drug? Or do you think it will be more like a single asset initially?
Yeah, so that's some IP I have. I envision it that one could do this much more wholesale than the retail kind of biology we're doing in my lab of 20 people. I think there are ways of thinking about this to get there thoughtfully over the coming decades. And I would turn around the whole idea, the reason we had Moderna and Pfizer mRNA vaccines so very rapidly is that mRNA technology essentially turned into, I don't know in Britain whether they're called the same things, but CNC cutters in machine shops. You just program it and whether it's laser, water, cutting, and that's essentially what mRNA technology is. And I envision some ways a reverse of that. In the purple.
Yeah, thank you very much for the talk. My question was sort of similar to that, double-clicking on the idea of circuit-customized medicines. My understanding is that potentially you're going to try and go after the different proteins that are mutated in different disease states, or potentially some of the RNA sort of molecular targeting machinery. I was wondering how that might play into that personalized or circuit-customized medicine aspect of it. So say you've got a diagnosis with this mutation, would you intervene as soon as you have that diagnosis, or would there, in order, especially with the development stage of it,
right?
You know, if the neurons have already migrated to the wrong area, which proteins are we going after? And
so you bring up a lot of sophisticated questions. We can talk about it over wine. We also don't know each other, but you're going to get a deeper view into my personality. One of the advantages or disadvantages of being a combination venturesome dreamer plus historian is it doesn't mean I need to be practical. So I think in practical details, you bring up a question, many mutations affect migration in addition to circuit building, which is why in that mTOR work I sort of glossed over, we made a mutation after the neurons had gotten to their correct place. And I think that's key. It's not just what one does, but when. And there are lots of evolving molecular tricks for timers and sort of time bombs or alarm sets so that you can make X, Y, or Z happen after a certain time. And I think these things sound wacky, but I don't think they're so out of the possibility in a decade. Professor Miller.
Jeff, thank you for the wonderful talk. And I also use exactly the same analogy when I teach about motor neuron disease, but I usually go all the way to Stansted Airport because I think the distance is a bit longer. So
I like to be conservative in my talks.
But you know, when these neurons, they start going towards Stansted Airport, they have no idea that they will go to the long-term terminal, on the third floor and the number 5 spot.
Yeah.
When they leave Oxford, all they know is I follow the particular signals, always the red signals. If you sit in a Toyota, you follow the red signals. If you sit in a Renault, you follow the other signals. So I think you over-dramatized the precision of these projections, because I think at the beginning they just follow very, very simple principles, and you identified with others many of these very simple turn right, left, at the initial signals, go to the callosum or go to subcerebral. Yep. So these are just 1 or 2 molecules, and then you have another set of molecules. So I think it's more manageable than how you presented it.
So Zoltan Molnar, and I'm going to tell you, I met Zoltan, I think, we have it down within about a year, when he was a finishing PhD student and I was a postdoc in about 1992. But I never over-dramatize Zoltan. But what I did do was simplify, but you're under-dramatizing it, because what we're finding, so there is something from insect biology of something termed guide-pulsed cells. And what Zoltan is pointing out is that when I start off my trajectory, I can't keep in mind the whole Google Maps instruction set. So I read step number 1. And first, I go out the door. At the door, I read the instructions that say stairway that way, and I go there. And it's been thought that neurons, in a sophisticated way, break their trajectory down, just like a 49-step route to Heathrow or Stansted. What we found out, though, is actually some of the instructions for later steps are already there in RNAs that are exceptionally long-lived, longer lived than most people have thought RNAs are. But there's an emerging biology you probably know about of circular RNAs and RNA granules that can keep RNAs stable for days. So these guys seem not to just know to go out the door. They seem to know, like many of us do with Google Maps, they know 6 or 8 instructions ahead. And they know I'm going to go out the door and down the stairs, but then at some point they get to a roundabout and then they do get some input. And we're very interested in that. So I think it's still pretty dramatic. And the fact that they know what to make to hear at the roundabout, that's remarkable. So, but Zoltan's exactly right. It's even more complex than that cartoon. But thank you.
Hi, great talk, thank you. I wondered about the localization of the RNAs and whether the signal that localizes them is within the UTRs, and if so, well, in either way, whether any of the variants that have been associated with the psychiatric or other neurological conditions are located within the elements that do the localization.
Right. So this is the next 5 years of work in my lab. We've just started that. And UTRs are untranslated regions, and there's one on one end, the 5' UTR, and one on the other end, a 3' UTR. We find there have been some global 3' UTR codes for being shipped down axons in a generic way. We found some other generic 3' and 5' UTRs that get shipped down axons and specific ones in 5', in 3', and we suspect in RNA conformation, because even though that central dogma slide has RNA looking like it's just a strand, they actually link around and make pretzels and curlicues. And so we're very interested that it's in both UTRs, in structure, in conformation, and in motifs within. For example, we identify, people have wondered in the nervous system, why are there something that some of you know about called so many splice variants? They must be redundant. Turns out different types of neurons use different splice variants. So there's splicing machinery. So we think this is a technical term, wacky wild. But that's what we have as a brain, that it's learned over evolution to send transcripts in remarkable ways. And we find some of them we think are maybe actively taken, and we're very interested that maybe at the axon hillock, maybe there's a filter that keeps some classes out, because it could be theoretically either way. We think some is trafficking, some might be exclusion, if that makes sense.
I'm assuming maybe differential polyadenylation as well as differential splicing.
Yes. We think all of these things, but this is just, and we're trying to, what I skipped over there is Priya Vairaghavan, one PhD student in the lab, is using something that you probably know about because it sounds like you might be an RNA biologist, SLAM-seq, to label with thiol groups with the genetically encoded UPRT so we can look in specific types of neurons what transcripts get shipped down and then do some mutagenesis and look at things like that. So that was not for the general historian, so.
So I think Professor Macklis has earned at least one glass of wine, but we have
Okay.
one final question over here before we.
Great.
So I'm a layman, and which means I tend to invest in these kinds of technologies once in a while. But I want to ask you about the general problem that, drugs are like sledgehammers, right? They act without a lot of discrimination. And I know of people working in synthetic biology, and what they're trying to do is to design logic circuits, right, that are protein-based that can be alongside with the drug and can go to a cell and can query it. What kind of cell are you? Are you cancerous? Are you this or that or that? And then decide if then else, destroy it or move on.
Yep.
Do you have any comments about that and how realistic this is and whatever?
The intellectual transfer office at Harvard has asked me not to go further into my thoughts in public, but yeah, I do have ideas about that. And I think it's way cool that people are making synthetic adapter molecules that can either block an interaction so that something won't, or make the interaction doable by changing conformation so something will. And I think there are all kinds of, I mean, it's like going on Amazon to look for doohickeys for your Mac. I think we're going to learn to make doohickeys that do various things. I hope the IP office doesn't mind the doohickeys. Yeah.
Jeff, thank you very much.
Thank you all.
I think the breadth and the depth of the questions is a good testament to how interesting your talk was. So thank you again, Jeff.
Thank you very much.